All right, let's go ahead and get started. My name is Abigail King. I'm a master's student here at Texas A&M studying enology. So they picked a wine person to introduce something that we can maybe use in the wine world. I'm happy to introduce Dr. Eric Anslyn, coming from our best friend, the University of Texas in Austin. He is a Welch Regents Chair in Chemistry and a distinguished teaching professor. He received his Bachelor's of Science in Chemistry at California State University, Northridge. He received his PhD in Organic Chemistry, focused on mechanistic studies of olefin metathesis. I hope I said that right. From the California Institute of Technology. He was a National Science Foundation postdoc fellow at Columbia, studying artificial enzymes. His expertises lie in mechanistic physical organic chemistry, supramolecular chemistry, and materials chemistry. Most recently, materials chemistry. His most well-known is the creation of new methods for chemical sensing, and we are so excited to hear about this new way to sense chemicals and the implementation in the wine industry. And give me 1 second. We have our second speaker is Diana Zamora Alvarez. She's an assistant professor of practice in the College of Natural Sciences and the associate director of the UT Wine Initiative. Her research areas focus in organic chemistry, analytical chemistry, biochemistry, medicinal chemistry, biomedical, and educational sciences. Her current position as research educator in the Freshman Research Initiative. She manages the supramolecular sensor streams at the University of Texas, Austin, leads undergraduate research labs, which involves mainly freshman student, but also mentors, and her lab develops pharmaceutical chemistry from the National Autonomous University of Mexico and her PhD in organic chemistry from the University of Texas of Austin. So I apologize. I should have introduced you as Dr. Diana Zamora Alvarez. Thank you both for being here, and we are so excited to listen to your wonderful presentation. Please take it away.
Well, thank you. Thank you so much, Abby. And thank you to Amit for inviting us to speak in this Texas A&M event. Yes, we're best of friends. I love going out to Texas A&M. Now, I will admit, I usually go to the chemistry department. I've never spoken. Am I speaking in a horticulture department, right? Yeah, that's pretty amazing. That's a first in my career. So I'm delighted. I'm going to talk about this, what was called new way of chemical sensing. And I'll start with this slide and make an analogy to how our senses of taste and smell operate. There's the lock and key principle that's taught in biochemistry, very often second semester of organic chemistry, where one thinks about an enzyme being highly selective for a particular substrate so that it can perform a chemical reaction on that substrate. So the lock, meaning the enzyme, fits very precisely the key, the one particular substrate. And lots of biological interactions are nicely thought of with that kind of an analogy, a highly selective receptor binding one specific target molecule. Well, our senses of taste and smell really are not operating this way. Instead, we have kind of what I'll refer to as sloppy receptors that respond to many different molecules. So in our senses of taste and smell, for example, in the sense of taste, we think about sweet, sour, salty, bitter, and umami, which means a savory, delicious taste. You know, many sugars elicit a sweet response. Many alkaloids elicit a bitter response. And there's not particular receptors, meaning our taste buds, sitting on our sense of, or on our tongue, that are targeting very specific molecules. And it's the same with our sense of smell. Instead of maybe 4 or maybe 5, 6 sensations with taste, maybe there's hundreds of sensations with smell. But still, there's not individual receptors except for potentially pheromones. There's still a bit of a debate, but there's not individual receptors targeting specific molecules. Instead, there's a variety of cross-reactive receptors that are exposed to a mixture of analytes that we register as an odor. And our senses of taste and smell are highly dominated by our sense of smell over our sense of taste, actually. So we started a while back to try to mimic this with organic chemistry. So we use plates, as shown here, where in individual spots in the plates, we have different receptors that are not selective, but instead are considered differential, which to me means they're cross-reactive. And you read a pattern out of the plate. And the pattern is very analogous to our patterning with our senses of taste and smell that give you the signature for a particular food or beverage. So where do we take this science? Well, wherever we can get funding is usually the honest truth. One is wine, and that's what I'm leading up to here. But early on, Diana, who got her PhD with us, who will be the second speaker here, studied cross-reactive peptides, where the peptides were fingerprinting, so to speak, kinases. Now, kinases are proteins that bind their peptide substrates and phosphorylate them. And many kinases are thought to be selective for specific peptides, but we had a collaborator, Kevin Dalby, who basically knew that the kinases are actually somewhat sloppy. They will phosphorylate many different peptides, or another way of thinking about it, a variety of different peptides can be phosphorylated by a variety of different kinases. So if we take individual peptide sequences and watch their phosphorylation event, where we watch all of them at once get phosphorylated, maybe we can register these fingerprints or patterns that tell you which kinases you have, or even which mixtures of kinases one has. And some of the kinases we looked at were ERK kinases, J and K kinases, P38 kinases, and these 1, 2 are alpha, beta, gamma, delta. These are isoforms of the kinases. These are supplied to us by Kevin Dalby, but we synthesize the peptides. Now, the peptides carry this very unique entity here that's known as a SOX fluorophore that does chelation-enhanced fluorescence that registers when a phosphorylation event occurs on a serine that's positioned proximal, or threonine here, positioned proximal to this fluorophore. So what do? We watch the fluorescence grow in as those individual peptides are phosphorylated by their kinases. But we're watching all of them simultaneously in a plate. So all the peptides are exposed to all the kinases, or conversely, all the kinases are exposed to all the peptides. And indeed, just like Kevin Dalby predicted, pretty much every kinase phosphorylates each peptide, but the peptides are phosphorylated to different extents. That's what's key. The different extents of phosphorylation allows us to create patterns of the phosphorylation events of the peptides that become diagnostic for the kinases. And this is one kind of patterning. This is called a linear discriminant analysis plot, where we can group, or cluster is the term used, the different families, J and K, ERK, or P38 kinases, but also separate even within those families to some extent the isoforms of those peptides. We can actually even follow concentrations of the kinases by watching these patterns here change as the concentration of the kinase changes. and then taking this kind of a plot and doing another kind of patterning that's known as a support vector machine machine learning approach, we can actually go in and quantitate the concentration of individual kinases in the background of all the other kinases that are present. Now, we may be getting pretty intense biochemistry here, and I haven't gotten to any wine, but what I think is the important take home from all of this is that we use cross-reactive peptides that fingerprint what are the kinases present, and then you can quantitate the kinases in a background of other kinases. We can also quantitate basically kinase inhibitors, because you may be familiar that a lot of cancer treatment is actually targeted at kinases that go apparent in their activity, and a large fraction of cancer drugs are actually kinase inhibitors. We're using these cross-reactive arrays of peptides. We can also quantitate concentrations of inhibitors, such as this MEK inhibitor shown here. Well, we wanted to expand the array of SOX peptides to try to get a better differentiation of all the kinases and their isoforms. And if you remember back, well, I can give you a quick reminder, there's really only 5 peptides here. We expanded our array to 14 peptides that are all, once again, cross-reactive, where our goal is to differentiate the isoforms within the JNK, ERK, and P38 families, where the kinases are given these terms here, and we're analyzing which kinases react with which families, which peptides react with which families of kinases. And you can see in this plot how cross-reactive actually all these peptides are. For these particular peptides, they're interacting with the JNK. The cross-reactive ones that interact with the ERK, P38, and JNK. Here's the cross-reactive ones that react with ERK and P38. Admittedly, these are just acronyms, but understand that they actually are standing for peptide sequences. Taking all of this together, our goal was something that nobody had yet achieved in the kinase field, and that was quantitating numerous kinases all simultaneously. So, in other words, in a background of all the components in a cell lysate, can we actually quantitate 3 different kinase activities as they go up and down in that background by using a cross-reactive suite of peptide receptors rather than relying on individual peptides, individual kinases, for each of the peptides. Highly selective receptors versus cross-reactive receptors is really what we're testing here. This is called a training plot, comes out of design of experiments software, where we're going to train on the various blue dots, which are mixtures of JNK, ERK, and P38, and our test plots are the red dots, and they are the test kinases, where we're going to train on blue and then see if we get the answer right in our machine learning protocols for the red kinase activities. And here's one single plot that shows, indeed, we can quantitate the 3 different kinases in the presence of the background cell lysates, as well as the other 2 kinases changing in their activity with about an 8% error. Now, one thing I've relied on in all of that I'm about to rely on in the wine analysis is what we call the patterning. If I can make another analogy to our senses of taste and smell, at some point when we're children, so to speak, we are trained to recognize all the cross-reactive responses that are coming out of our senses of taste and smell. For example, at some point, we're told you're eating chicken broth. At another point, you're told you're eating beef broth, vegetable broth, etc. And it's all the patterns of our senses of taste and smell that get imprinted in our mind and associated with a particular food or beverage, beef, chicken, vegetable, in my kind of silly analogy. Well, we need some kind of patterning that the computer would use to recognize the responses from all our cross-reactive receptors. And the patterning comes out of field of chemometrics, now commonly also referred to as machine learning. And we particularly love principal component analysis and linear discriminant analysis. And I'm going to show you some of those plots. I'm sure Diana might also in her talk, where you're looking at scores. And if the scores are closely clustered, but widely separated from one another, that's indicative of the fact that you've done good differentiation. And if I may go back for just a second, that's exactly what we looked at here, where we were able to score the different kinases, but also even subtly pull out some clusters within each class of kinase when we looked at their isoforms. So I want to take this whole concept forward of machine learning, chemometric analysis of many different analytes in a complex mixture and start to analyze wine. Now, we could say, why did we choose wine? Of course, I'm talking to a department where wine is of interest. I chose wine simply because I thought it was fun. I mean, literally 22 years ago or something when we started all this, everybody loves wine, right? I'm probably singing to the choir here. So it was a test bed set of analytes that most humans can relate to. And they can relate to people like this gentleman, who's a sommelier, who can tell you all these taste discriminations about wine that he implies you should think about your wine. So of course, there's people who make a living tasting wine and reporting on its quality, and even reporting to you what you ought to think about that wine. Now, they've highly trained their palate to be able to pick up on subtle differences. Can I train a suite of cross-reactive receptors in a 96 well plate to pick up subtle differences in the wine? Absolutely identically to how we trained a cross-reactive suite of peptides to pick up on differences on kinases. So this is in collaboration with Hildegard Hyman, who's a professor of enology and viticulture at UC Davis. I certainly got introduced to her a good couple of decades ago, and she helped us set up these studies. And what we train on or pattern are the tannins in red wine. Here's a smattering of the tannins we're all drinking whenever we drink a red wine. As an organic chemist, I focus in on the fact that they're catechols and resorcinols, functionality that we teach terminology of in organic chemistry. A lot of them are glycosylated and carry sugars, like this one down here. So if we can make a suite of receptors that will bind every single one of these tannins, but in a certain sense, those are sloppy receptors, meaning they bind every one of these tannins, but bind them differently, maybe we can fingerprint these tannins. The way in which our lab does this is create receptors out of peptides, where the peptides are highly biased towards having histidine in their background. And histidine is the side chain of, imidazole, is the side chain of histidine. So our peptides are heavily biased towards histidine, such that they bind a metal and then bind a pH indicator. And because many pH indicators carry catechol functionalities, the same functionality that's in a tannin, if you incubate these peptides with tannins, you'll displace the indicator that's blue and it turns yellow. And voila, you got a very simple colorimetric assay for the tannin. Now imagine having a variety of peptides with a variety of metals and a variety of pH indicators, all of a sudden you have a 96 well plate that fills up with a series of cross-reactive sensors that all respond to tannins. This is done in an undergraduate lab at UT Austin. This is what Diana will spend a large fraction of her portion of the talk discussing. Here we have some names of undergraduates. This is not done in my graduate research group. The kinase work was done in the graduate PhD granting program that I run. This is done with a bunch of undergraduate programs that Diana supervises, where the individual students get various peptides that you can see over here in the single letter terminology of amino acids, the sequence of the peptide given with these single letter amino acids. And then they synthesize these and they start to learn how to do the indicator displacement assays with the metals that give us the colors. And we process those differences in colors with linear discriminant analysis, as I discussed, one of those score plot techniques. And one of our earliest samples showed that we could immediately discriminate a Cabernet Sauvignon from a Zinfandel, from a Merlot and Pinot, and you can see here, we generated a score plot that differentiated the different varietals in 2 dimensions or in a 3-dimensional score plot. Well, there was an outlier in this early study. Here we are differentiating a variety of different varietals where we clustered together a bunch of Zinfandels, and indeed notice the Zinfandels that we bought all seemed pretty similar to one another. There's subtle differences, but they were pretty much all clustered together because a Zinfandel is a Zinfandel to some extent, even though the root stock may be different, it's grown in a different site, so on and so forth. But I bet most of us can taste a Zinfandel and know that it's a Zinfandel versus these other red wines. It is distinct. But there was an interesting outlier. And I had no idea why there was this outlier. This over here is purportedly a Zinfandel. Well, I got a phone call, and it's not a Zinfandel. It's a Pinot. And I want you to note where my Pinot Noir pattern was. It's the triangles. After the fact, we were told this is actually a Pinot Noir, and notice it's pretty darn close to the Pinot Noir. So in other words, there was a falsification on the label of that wine that someone called and told me. Well, this actually led to a series of analyses of falsifications of wine, in that there was a class action lawsuit in Northern California against Constellation, which owns a lot of wineries, as well as Gallo, because Constellation and Gallo were selling Pinot Noir wines that were actually Gamay. And the way this worked out was they had been falsified themselves by the importers, where the importers from France told them they were Pinot, but they were actually Gamay. But still, Constellation and Gallo sold them under the label of Pinot Noir. Well, they were caught, and fake Pinot Noir get a break from, it made Reuters news, it made national news, this class action lawsuit. And the best part of this class action lawsuit was the fact that with respect to the Constellation Net Settlement Fund, meaning the payout that Constellation and Gallo had to do for losing the class action lawsuit, the University of Texas at Austin and Professor Eric Anslyn's research, I got a grant in basically. Due to a lawsuit, this is the only time I've ever had a grant that's due to a lawsuit. I wish there was more of this happening, where this is now funded the FRI program that Diana will be discussing for quite some time, a lawsuit supporting this undergraduate research lab that's trying to differentiate varietals of wine. Well, we went on further with Hildegard Hyman to not only differentiate varietals, but to differentiate blends like Cabernet Sauvignons mixed with Cabernet Franc and Merlot. If you remember in my kinase work, we were able to differentiate mixtures of ERK, P38, and J and K kinases. It's perfectly analogous here. We're going to differentiate mixtures, blends of Cabernet Sauvignon, Cab Franc, and Merlot with a training set and then some test sets. And we get these plots that allow us to train with the red dots, come in with test sets, which are the black dots, and see how well we can quantitate a blend. So our blends had a reasonably large error, but still it worked to pretty good extent. 18% error in whether we could differentiate 18% error in the Cabernet Sauvignon, 15% error in Merlot, 16% in Cab Franc. Meaning I could tell you 46% Cab Franc plus or minus 16% or so. So fairly large errors, but still it's working. But it's a lot better than a bunch of chemistry professors because I had a fun experiment. I got my faculty together and we compared the faculty against our device, and the faculty could not at all appropriately classify what was the varietal mixtures of Cabernet Sauvignon, Merlot, and Cab Franc. I don't really know how many humans could even do it within a 15% error. So I really want to, oh yeah, I got one last thing, but I really want to hand this over to Diana. We can also differentiate regions that wines are grown in Northern California and Southern Oregon, the Willamette Valley, Arroyo Seco. You could see these various valleys. We're able to now classify the wines as to where they're grown by valley. But I think it's really time to pass the torch over to Diana and let her show what our undergraduate laboratory is now doing. So I'm going to end here and let her take over the show because I went a good 2 or 3 minutes longer than I wanted to. Diana, I'll stop sharing. Beautiful. So I can't hear you.
Here I am.
Yeah.
Can you all see my screen? Okay, perfect. All right. So thank you, Eric. We wanted to give you an overview of the current applications of our differential sensing workflow. And as Eric mentioned, like the idea is that once we have different type of wine samples, with the peptide-based sensors that we synthesize in the lab, my students are able to actually execute the assay. And by using, a regular well plate reader and computational analysis, chemometrics, or machine learning, we're able to classify wines according to varietals, blends. We have other studies that show also the hang-on time on the vine, the year of harvest, and finally, different vineyards or regions. This type of work has been previously been done in collaboration with professors at UC Davis. And we decided to switch gears and started a strong collaboration with the wine community. So that's where the Wine Initiative program within the College of Natural Science started. We started this program a few years ago. And as part of the differential sensing assay that Eric described before, my students recently have been able to work with wines that are supplemented by wine industry collaborators. And for this study, we use different type of Texas wines from different vineyards. And remember, here we're looking at a specific fingerprint that each wine will display according to their tannin composition. Over here, we have one of the principal component linear discriminant analysis plots that Eric described before. And what it's interesting to observe is that we have different varietals coming from different regions, different vineyards across different areas of Texas. We have wines from the hill country within like Fall Creek, Salt Lake, and also we have all other wines coming from other viticultural areas such as Mesa and Cereberg. So what we found is that, again, the differential sensing method that we use is pretty robust in classifying the different wines. So we were able to classify and differentiate the different varieties. And interestingly, once we label the varieties according to their respective vineyards, we're also able to observe a classification of those vineyards. And we can observe that Salt Lake differentiates, strongly from Mesa and Cereberg, whereas Fall Creek is somehow, like in close proximity to some of the Mesa, in between Salt Lake and Mesa. So within this classification, we are in general exploring how the wines from Texas, they classify within each other, the different varieties, but also according to vineyards that are like in Central Texas and in other different areas. And so with this study, we were able to classify Texas wines. And one of the common studies that we're currently working with is to also characterize the metabolomic profile of those wines and complement that analysis with the previous linear discriminant analysis. So now we are also working with the analysis of Texas grapevines. Specifically, we're working with the analysis of the leaves and the grapes. And for this study, we harvested samples from different vineyards within here, the Texas Hill Country, and again, from Cedarburg and Mesa. And we decided to work with about the same varietals from that will correspond to the wines. So so we can continue, we compiling more information about just the general samples like wines, but also grapevines, and see if we can find any correlations. So with this, with this type of samples, what we decided to do, because, the differential sensing approach that we have previously used, it has focused mainly on detecting tannins and classifying the wines based on that tannin composition, that it's very unique to each of the varietals. So for us, in order to tackle this type of analysis within the grapevines, leaves and berries, we decided to switch to a different type of analytical method. So we decided to start working with metabolomics. And metabolomics is part of the omic research efforts that, metabolomics complements other areas such as genomics, transcriptomics, and proteomics. Here specifically, we're studying endogenous metabolites, low molecular weight molecules that are either precursors or byproducts or metabolic pathways, and that they specifically provide information about the functional status of grapevines in this case. Now, I think, can all agree that grapevines have a very rich chemical diversity of different classes of metabolites. And there are some studies that they have trying to correlate that chemical diversity to their adaptability, but also to the type of unique metabolite features that are present in those grapevines according to the region that they are grown. So it's also understood that the phenolic composition of grapes is genetically driven, and it's greatly affected by environmental factors. So as part of different meetings and brainstorm discussions that we had with our collaborators in the wine industry, our goal was to see if using metabolomics we could identify what metabolites are uniquely present in Texas grapevines, but also if we can observe a difference in their abundance. So there are a couple of studies that they have been, in the last decade published in other universities where there's this collaboration between the wine industry in certain region and different institutions. Where they are able to identify different classes of metabolites that we know they are responsible for the aroma and the sensorial effects of wines. So for instance, we have flavonoids, phenolic acids, silvanoids as part of these different classes of metabolites. And those are the type of molecules that we aim to identify, and we aim to use them to classify the samples that we're working with. For metabolomic profiling in general, there are different stages. Within this study, we decided to study 17 different varieties coming from 5 different vineyards. And after we harvested all the different samples, also considering that we harvested during the growing season, June, July, and August within here, the Texas growing season, we perform a 2-phase extraction of the metabolites, where we're able to obtain different a polar and a non-polar phase. And the secondary metabolites, which contains mostly the polyphenols.
May I ask a question?
Oh, yes.
Yeah. Before you get too far, make sure I'm not lost. So what you're doing with this profiling is establishing some, you were calling it a fingerprint with is that what you're doing with these? Is that the purpose of this particular process, the profiling, is just to get a base?
That's correct. We're trying to, yes, we're trying to obtain a baseline of the metabolite composition of the grapevines, and that is present in the samples. And within this process, so this is part of the experimental workflow. And once we are able to process all the samples, we have to work with a specific data analysis pipeline within the metabolomics methods. So some of the results that we have been able to obtain, these are some of the preliminary results that my students were able to obtain. In here, this is an example of 3 different varieties coming from 2 different vineyards. So we have here Mourvedre and Sangiovese and Tempranillo. This classification, which is shown again, like there's a difference between the different varieties, is it's due to the presence of different metabolites. And whereas, with the previous fingerprint method that Eric showed, where it's mainly based on the absorbance changes displayed due to the different tannin composition or unique tannin composition. In this case, the classification arises due to the differences in the metabolite content. And part of the analysis in here is displayed as this heat map, where in the x-axis we have the different classes of different varieties. And on the y-axis we have the metabolites that were identified using mass spectrometry. And within those metabolites, we can observe that there are already different classes of metabolites. We can observe here different flavonoids. There are some lipids as well, but most of them contain different classes of flavonoids. And what it's interesting to observe is that their abundance relative to each other is different as well. So the red squares indicate a higher abundance of those specific metabolites, whereas the blue color represents a lower abundance. These are preliminary results that my students were able to, we process the samples, we perform the mass spectrometry analysis, and then the data analysis pipeline. So this shows some of the preliminary results that we are able to obtain using metabolomics and how we can use metabolomics to classify the samples. So again, yes, we're using this method, these technologies, to establish a baseline and to understand what are those metabolites that are prevalent, that are present in the samples, but specifically that contribute the most to the classification of these samples. And in this, part of this study here, we were able to process all the samples. So in here, I'm showing all the different 17 different varieties, and where the varieties with their corresponding vineyards. And over here, with the principal component analysis classification, we wanted to first just observe if we could, observe any differentiation or classification at all. And it's important to remember that PCA is an unsupervised method, so it doesn't know, the algorithms, doesn't know what type of class or label was assigned to a specific data set within the analysis. So this is a raw type of classification, and it shows, some clustering within the different vineyards. And here we're looking at the classification of the vineyards. And here, in the 2D plot, we can observe kind of like a tight sort of overlap of the different clusters. And when we look at the 3D plot, which includes an additional axis of variance or differentiation, we can observe that classification and differentiation is a little bit more spread. So that gives us confidence in moving forward, so then we can use other methods such as PLSDA, which is very similar to LDA, what Eric mentioned before. LDA and PLSDA will focus on maximizing the separation of the different classes, or in this case, the different vineyards. So we can observe a better separation amongst them. And again, what's interesting in here is that the samples that we collected from Fall Creek, which are these two in here, they are the ones that scatter the most, they spread the most. And, we are currently looking into the data to understand what are those metabolites that are contributing to this specific type of classification. With this, this is my last slide. So I just want to thank you and just mention that, so far, these are the steps that we have been able to accomplish with my students. And we are currently in the process of finishing the analysis and also specifically through trying to identify the key metabolites that contribute significantly to that type of classification. That was an example of the June samples that we collected. So we still need to analyze the samples from July and August. And we're hoping to observe change in the metabolite abundance and potentially the content of those, specific varieties across the growing season. So that's part of what our future work is part. So with that, I want to thank you all, everyone who attended the seminar, and Eric and I were happy to take any questions.
Well, thank you very much. This is very exciting research, and I already have a few questions in the chat. And the first one is from Gary about the differentiation of the faculty who could not distinguish between the wine. But what about the sommeliers? Were they able to distinguish?
I didn't have any sommeliers in that test set. So I can't tell you at all. Thank you.
All right. Well, the next question is from Andrew. We also call him Alexander, I guess. So his question is, it's very fascinating, and he's very, we join him thanking you for sharing this. This could also, you could create a complete sensory profile for each varietal. Do you plan on trying this on white and rosé wines as well?
So yeah, we've tried white, and we really need to go to a different set of analytes. And, naturally so, there's just not many tannins in white wine. And really the receptors we constructed, we designed them to be cross-reactive to tannins. So on some level, it was a little bit of a relief that the method didn't work on white wine, because it helps to prove the premise that we really do need all those tannins. But if I think of another class of analytes that would differentiate white wines, yeah, I'm of course always looking for another project.
That's great. May I ask a question as well? It kind of leads off from what you just made a comment on, Eric. If there were, if you wanted to really develop a new receptor, so to speak, what would it take? I mean, of course, I don't know if it's an organic chemical, of course, and which contributes to some faults, perhaps, in the wine.
Right. So what is our method for making a receptor in a certain manner is what, yeah. Okay, so this is the field of referred to as supramolecular chemistry. That's a bunch of organic chemists sit around and we design receptors for analytes. Most of the time, our receptors aren't incredibly selective. And in fact, that's going back a few decades, what inspired me to mimic our senses of taste and smell, in that our senses of taste and smell aren't very selective either. And here I've been working with all these receptors that aren't very selective. Let's see if I can exploit them in a totally different context. And that's this cross-reactive array idea. I remember one of the very earliest grants from the National Science Foundation. I had a reviewer that said, wow, he's making lemonade out of lemons, which is pretty spot on. So to make a highly selective receptor, supramolecular chemists aren't very good at that. I'm being honest, Amit.
Sure.
Biologists are good, biochemists, they'll make an antibody, get a biological response to be able to make a highly selective receptor. But organic chemists still, even in 2022, aren't particularly good at this. But when we attempt to do it, it's a lot of computer modeling, making models, seeing if we can actually synthesize the target, synthesizing it, testing, and cycling. synthesis and test to optimize the receptor.
Got it. Thank you. Here's another question from Katie's iPad. Can your methods of sensing be used early in the process so that winemakers can use the data to improve their wines? Fascinating topic. Thanks.
Yeah, I suppose so. But what you have to do, so the answer is surely yes, if you've been able to train on what you call tannin distributions, or any other chemistry for that matter, of what you consider as a good wine. So if you have what you consider a good wine and you've trained on it, and you're trying to take a wine to that particular mixture of tannins, that is quote unquote the good wine, I suppose you could use our technique early on to see if you're heading in the right direction or not. Are you heading right towards the mixture of what we call good tannins, or moving away from it? But it always requires you to have a training set that knows what you're trying to achieve.
All right. Thank you.
Hopefully that
Here's
makes sense.
another question from the audience.
Hi, and this question's for Diana. And I really enjoyed your talk today. So I thought it was kind of an interesting finding that things were grouped by location rather than by varietal. If maybe I'm not understanding the study quite right, but what would be the underlying sort of reasoning for that? What do you think that's going on there that's driving that?
Yes, thank you. And just to understand better question, is that referring to the classification of the Texas varietals, the wine varietals and the vineyards?
Yes. Yes.
Okay. Yes. So, like when we perform this type of analysis, I think it resonates with Eric's thoughts in the sense that first we're trying to just to get a sense of like what the classification of the wines would look like. And usually they tend to classify according to, like it could be to the varieties, right? Like, so if they are, we have more than, different type of varieties, they will classify within themselves, or they will tend to classify by regions. And I think that you probably are thinking about the Lenore versus the Cabernet Sauvignon, like which come from the same vineyard, but they classify like opposite or separated from each other. So it's hard to tell with wines what's the main responsible factor for this type of classification, right? Because we have, with this type of studies, we have learned that it's not only dependable on the variety itself, but a lot of other factors such as the region, the soil where they are grown, will influence the development and the content of those tannins. So we don't want to have we don't want to say the final word that, in the sense that it's due to this or that reason. There might be different factors that are contributing to these results. So we will have to further dig into, what's going on in there. So yes, I hope that helps, with your question.
Well, just a little follow-up on that. So you're saying that it could be something about the winemaker at that particular winery rather than the location of the grapes and what's in the grapes, or
right. So
okay.
that's yes, I agree to that. So the end product, the finished wine, which it seems to be influenced by several factors like environmental ones, but also through the winemaking process, right? So we don't know which one will specifically contribute the most to it, and that's where we will have to dig a little bit further. But I do believe that part of the winemaking processes will, definitely, like since some of the tannins or polyphenols are developed during those stages, right, the fermentation stages, or they change, they become more complex. Those might be factors that may influence that type of classification.
That's great. Thank you. Any other questions? You can unmute yourself and ask the question or type it in.
Yeah.
Yes. Go for it, Abby. I'll go
Okay.
after you.
Do you see this research being used for kind of the establishment and also the confirmation of American viticulture areas, being able to establish those that you saw, I think, in Dr. Anslyn's when he showed the map of California and Oregon? Do you think that you'll be able to create enough sample set in order to distinguish different AVAs?
Yes.
Go ahead. Yeah, please.
Okay, yes, that's correct. And that's actually one of the studies that it's right now under development in our lab. We are, part of our efforts, it's to, like our bottleneck is to actually reach out to all the different viticultural areas, right? Because it's just huge, right? Texas is huge. And having those, collaborations and samples, right, through our collaborations, get the right size, of samples, so we can have a representative sample pool of those areas. That's what we are looking for. So we're in the process of gathering those samples so we can extend the study not to just specific vineyards, but to all the different viticultural areas, which I think it will be very interesting to observe, like how if we can classify them based on the areas, and within the areas, maybe according to their specific vineyards as well.
Absolutely.
Great.
That sounds awesome. Thank you.
Thank you.
Alexander.
That's funny. That's exactly what I was going to ask about. Like, I was going to take a Cabernet, for example, and see, could you distinguish a Cabernet from Texas, from Sonoma County, California, and let's say Southern Oregon?
I think that,
So very similar question.
and I think that is possible. In the sense that the Cabernets are going to be different, and yeah, okay. But it's a good question. Will we always be able to consider every single Cabernet only from one of those regions, irrespective of the rootstock and irrespective of the winery? I don't know, is a good question. In the one study, I probably didn't make it as clear as I could have, where we were looking at regions. They were all Pinot Noir wines. They were all the same rootstock, and the winery was UC Davis. So there was no variability in the winemaker or variability in the, the wine genetics itself, because they all came from the same rootstock. The only variability was the region of Northern California and Southern Oregon. If you start mixing all of that stuff together, I don't know what was going to happen. I think that's a fair answer.
Or
Awesome.
question.
Thank you.
Yeah.
Any other questions?
I have another question
Yes, please.
that's sort of a follow-up to my chat question. I'm Katie's iPad. There was a person behind it. And Abby's and Andy's questions, which is basically that wine is a very, very natural product at this time. There's very little that they're allowed to do to it to alter it. I mean, they do everything they can, but there's just some lines they don't cross. But if a winemaker got what he considered, like you said, the very best wine and ran your process on it, and then got a mix of what he thought was the profile for the very best tannin, then couldn't he artificially produce supposedly great wine, and couldn't you be the impetus for making wine an artificial product in the future?
Actually, yes, if I may,
Please,
a few
Diana,
years, actually not long, a few years ago, we actually started testing some wines that they were called replica wine. And I don't know if you were, like you came across them at some point, but these are wines that they're supposed to replicate, like the sensory, like the flavor of like really expensive wines. And they were pulled out of the market, like later on. But we were able to get a hang of them and test them. And we observed that, yes, like like just according to the clustering and where the clustering fit, some of them did fit where, like if they were saying like, oh, we have, we're on an analogous, like Pinot Noir or like a very expensive, like Zinfandel, they will claim a specific brand. So we actually bought the brands, and the replica, and just like regular control wines, and just check out like on the regular clustering. Some of them, they did fit within that claim, but some others didn't. And so it's like that's straight to your question. Like there might be people who will probably try to do that. And I think, like for us, it's not like, we're not trying to demonstrate if someone is right or wrong, right? Like for us, it's just like, it's fun. It's, like the students love working with wine in general. And it's an amazing tool to learn. But it's one of the outcomes that we're able to demonstrate, just like Eric mentioned with the lawsuit, like the class action lawsuit, like the technology or the method is very sensitive, that we are actually able to classify and observe those differences.
Thank you.
Thank you.
One more question maybe, if there's one. Well, there's a comment for Eric and Diana to see. Our professor Patricia Klein's daughter was a UT grad and is a UT grad and participated in this FRI program. So, and that's pretty cool. So yeah, we do have a good long history of interacting with you guys through our faculty. And Eric, and Diana, since there are no more questions, it seems, I just want to say that every time I interact with you, just amazed and impressed beyond words of this kind of cool work that you're doing over here. So thank you to both of you, and we really are grateful that you presented, and hope to host you in person one of these days when time works out.
Thank you so much.
Thank you so much, Amit. It's our pleasure.
Yeah.
Thank you so much,
Absolutely.
everyone.
Thank you, Amit. Thank you to everybody.
Thank you. Bye-bye now.
Okay, bye-bye.